So, welcome back to Office Hours Unplugged, where we chat with the most prominent researchers and PIs here in McMaster. Today, we will have an office hour with Dr. Sara Andres joining us. Thank you so much for being here. Could you please introduce yourself to the audience and let them know a little bit about yourself?
Well, thank you both of you for having me. I really appreciate the opportunity. So I am an associate professor in the Department of Biochemistry and Biomedical Sciences, which is within the Faculty of Health Sciences. And I'm also currently the director of the Biomedical Discovery and Commercialization Program. And my research interests, I really like understanding mechanisms of DNA repair using, structural biology and biochemistry to answer my research questions.
And how did your journey through that whole thing start? I mean, your journey through academia in general. I mean, where was your starting point?
It goes all the way back to high school. I took all the classes you're supposed to take, and science was just the one that I really loved. And I'm old enough to I was taking OECs, which is essentially grade 13. And OEC biology was really biochemistry, and that's where it really clicked. Seeing the Krebs cycle and metabolism and how these enzymes convert molecules to energy was really what hooked me, I would say. And so then I went to the University of Guelph for my undergraduate and took biochemistry there. And so I kind of knew, okay, science is great. Let's see where it goes. And I'm sure a lot of you going through undergraduate, what do I do next, right? And so my mentality was always, can I challenge myself to get to the highest point in that field? So for science, like academics, right? Okay, get your undergraduate degree. What's next after that? How about graduate school? Now that it's graduate school, can I become a professor? So that's sort of been the mentality along the way. I wanted to challenge myself, but I knew I also wanted to keep some element of teaching in there. So whether that would have been going off to be a high school teacher would have been great. My mother was a high school teacher, so I had that, guidance there.
Wow.
But then when I got into, graduate school and stuff, I saw being a professor could be kind of a really cool place to go. So that's sort of how I went. Yeah.
And is that kind of what drove you to do research? When you saw professors, were you more interested in the teaching field, or were you more engaged or interested in the research field?
That's a really good question. To be fair, when I was an undergraduate, it was probably more the teaching side of things. My fourth year thesis project, while a good experience, I didn't love it. I'd spent a lot of time in a cold room making lipid vesicles, and I'm like, it wasn't that exciting, but at least I understand why. It was a good project for an undergraduate student at the time. But I think the thing that really hit me is I went on graduate school tours, and I came here to McMaster, and I went on a tour of the structural biology facility, and I'd never seen a structure of a protein at that point. I was in third year, so it was a little early, and I kind of saw that structure, and I'm like, that is what I want to do for the rest of my life. I love this idea that you can see how the carbons and the nitrogens and the oxygens come together to form a protein, and that gives you some insight into how it functions. And so, I went back to Guelph. In my fourth year, took a structural biology course, and that sort of got me on the path to focus, more on research as well, but also with some emphasis on teaching. Yeah.
So you didn't really get into all of your structural biology interests until grad school?
Yes, exactly. Yeah, it was a steep learning curve.
Oh, wow.
Yeah.
And where did you do grad school?
I did it here at McMaster, actually, in biochemistry. Yeah, I've been really fortunate to come back.
Okay. And what was the most challenging part of your academic journey? I know, that learning curve you mentioned must have been really difficult, but was that the most challenging part, would you say, or was there something else that you found particularly hard?
The learning curve at the start of the master's was probably the most difficult thing I've ever encountered. So undergraduate for me, there were challenges, but I was able to learn it. I could get through it. I did okay. But then when I got to the master's, everything you touched failed, right? I wasn't used to that failure constantly. Research is 90% failure or higher, right? And so I was so done at one point. I was like, I'm going to get the master's and go to teacher's college, and that would have been fine. But it was really because I had a PhD supervisor who just kept encouraging me to keep trying things and let me make mistakes. For example, I don't know if you know what an FPLC is. You're in biochemistry, so probably,
FPLC,
fast protein chromatography. It's basically an instrument that allows you to purify proteins. Instead of a gravity flow column, you can put on a machine and it does the work for you, which is nice.
Interesting.
That thing terrified me when I started graduate school. There were so many tubes running everywhere and so many buttons to hit, and it was very finicky. But by the end of my PhD, I could take that thing apart and put it back together. So, it was
amazing.
Yeah, scary at first, and that failure was tough, but you get through it.
Right. It helped you grow in the end, after all, right?
Definitely. Yeah. I learned that making mistakes was not the end of the world, right? It's a good thing. You learn from them. And you learn them from better sometimes, I think, because it really sticks with you.
And throughout the whole experience, did you take any lessons that came back to help you when you became an independent investigator, like a PI? Was there anything that you learned throughout your grad school experience that kind of, oh, I think this is how you're supposed to run a lab? Like,
yes, I totally think so. There are things that I saw that I liked and things that I saw that I didn't maybe like as much throughout my PhD and my postdoc experience. And I took all of those tools. The big one being making sure students were aware it's okay to fail. It's okay to make a mistake in the lab. That's totally fine. That's normal. That was one of the big things. And just making sure that people always knew that I was there to help them out, right? That, and also asking your friends for help, asking questions. That's a big one, right? If you don't know something, find someone who knows it,
right,
or knows better, right? And I was always very much like in my corner doing my own thing, and I needed to open that up a bit. And I got better throughout my degree, and now it's like very much, if I don't know the answer to something, there's somebody who does, and I'll find them and they can help out.
Okay, that's amazing. And you mentioned you had a really good PI who encouraged you throughout your graduate school and your graduate studies. So when you were first creating your lab, was that person an inspiration to you, or was that like the person or the type of PI you thought you wanted to be?
He definitely was. So I should mention his name. His name is Dr. Murray Junup. He's now at Western University. I loved that he let you explore your options. He never really confined you to anything, right? If you had an idea, he'd say, go for it. He was always super encouraging as well. He was always like, Sarah, you can run a lab one day. Because I was like, I don't think so. And he's like, yes, you can do this. If I can do this, you can do this, So that was one thing. And also, I really liked the environment he had. When I started my lab, one of the things I really wanted was to have like a good team community vibe in the lab, that people would feel comfortable talking to one another, bouncing ideas off of one another. And so that sort of helped me start my own lab and led to the inspiration for that.
And going into your lab, what was the original vision for the Andres lab when you first started out?
Yeah, the original vision, of course, was that community team mindset, right? Because, research is hard, but it's the people that really make it fun. And so I think that was important. And I want it to be balanced too, right? Like I want people to love science and do their science, but I also wanted them to be able to have hobbies and a life outside of the lab. It's super important, right? So when you have all those failures, you have something else to, bring back your, bring back some life to everything. So that was important. And I knew that I needed to start with what I knew, right? You don't want to go into your own independent lab and try something you've never done before. I mean, you can, but maybe wait a little bit. So I knew it was going to be a structural biology, biochemistry focus because that's what I knew.
And did you know you wanted to do it in McMaster? Because you mentioned you did your graduate studies in McMaster, right? So were you thinking of different universities, like Western, maybe U of T, Guelph? Or were you kind of dead set, like, I want to open up my lab, I want it to be in McMaster?
Choosing where you to have your lab is tricky because there's not a lot of opportunities to become a professor, right? It's tough. There's limited space in Canada. The dream was somewhere in Southern Ontario. McMaster was sort of the number one choice. It was a number one choice because I knew the resources here. I knew what they had for structural biology, biochemistry. I knew the people here, like the professors that were there, and I thought that would be an amazing place. And also for me, geographically, it was ideal because I have family close by and I wanted to be close to them because that's important for my life and my well-being. So yeah, when the position opened up, it was like literally a dream come true. I was like, I need to get this job. I've got to do everything I can to get it. And honestly, the day they told me, like, we're going to offer you the position, I couldn't believe it. I'm like, are you sure? Is this true? I'm not dreaming? So, yeah, it was amazing.
Oh, that's awesome.
And going a bit more into the technical side of things, can you talk a bit about the equipment you have in the lab? Because I was doing some reading, and you have some really cool toys that you can play around with.
Yeah, that's perfect. I do refer to them as my toys. My goal is always, what new toy can I get for the lab now? So for structural biology, I'm an X-ray crystallographer by training. And so the great thing is that I inherited the X-ray diffractometer that was already here from the previous structural biology people. So basically, it's an X-ray generator that then focuses the beam into a really fine beam that you shoot at a protein crystal to get your diffraction pattern. And so I've got that piece of equipment, and I was really fortunate to upgrade it a couple of years ago. So we've got a really fast detector. So the old detector was super slow, and it would take you, I don't know, 8, 12, 24 hours to collect a full data set. Now you can get that in like a matter of like an hour or two or less.
Wow.
It's amazing. So I love that. I also really love the liquid handler robots we have. So if you've ever done crystallography before, as a graduate student, I set up everything by hand. It's very repetitive, very boring. And so I'm really happy that we have robots that can set up a crystal tray now for you so that no one has to get carpal tunnel syndrome. So that's great. And then we have an automated imaging system. So you can take your crystal tray once the robot set it up and put it in a box, if you will, and the robot will automatically check your tray for you every, however you set it up, every 24 hours, every 3 days, whatever you want. And then you can just look at a computer screen, which might be annoying, but let me tell you, it's way better than looking through a microscope at, hundreds and hundreds of drops of protein crystallization trials.
Wow, that's amazing.
So those are fun.
And for anyone that doesn't know, the liquid handler robots, they're just like a whole array, basically, of a hand that goes in and just dispenses whatever you want in there. You can customize it.
Yeah, exactly. Yeah.
And you think those really expedited the process for you?
Oh, it makes everything so much more efficient. So by the end of my PhD, I could set up one screen of 96 different drops of trials in 30 minutes. Wow. The new machine can do it in about 10, and it's much more consistent, right? It can pipette volumes as small as, I want to say, it was 25 nanoliters is as small as it can go. The smallest I could do with a handheld pipette was like 1 microliter, and even that was only as accurate as your pipette was. So, it's pretty cool to have that. Yeah, ours has, so every liquid handling robot is different. Ours has like a set of 96 pipette heads, pins basically, that put your screen in, and it's got a little a nano needle, it's called, that picks up your protein and then spits it out back and forth across the tray. It's pretty fun to watch.
That's amazing. Yeah. And just kind of going into more general questions. So I know you have many students in your lab. So is there any project in particular that you're really interested in or looking forward to seeing the results of?
Oh, it's like you're asking me to pick my favorite child here.
Just the most interesting one. Just the most interesting.
Yeah. So I've got one project that's newer on translesion DNA synthesis in bacteria. And different bacterial species use different proteins for this. And there's one set of proteins that have been studied really well. There's another that haven't been studied as much, and there's like no biochemistry, no structural biology because there's some technical challenges with it. And we've been really lucky to overcome those technical challenges. And so now we've got really cool results. We're finding out new things about the protein and how it functions, what some of its enzymatic capabilities are. So I'm super excited to see how that goes. And then one thing I haven't talked about is that from a structural biology perspective, I'm an X-ray crystallographer by training, but there's another technology called cryo-electron microscopy. It used to be not as popular because it was more blobology. The resolution it would give you blobs, not the atomic resolution.
Right.
But then there was this huge jump in technology where the detector is so good now you can get like direct electron detection and you get that high resolution that you need for an atomic structure. And so we've got a project where we're starting to get into the cryo-electron microscopy through collaborations and through sending my students to learn in somebody else's lab. And there's some promising grids there. So the grids are what you put your sample on to get the data. And so I just need to go back to the lab right now and book some time at a cryo-electron microscope to collect the data and see what we have.
And can you speak a little bit about that aspect of spectroscopy in general and how it's developed quite rapidly in the last decade?
Yeah, for sure. I mean, I wasn't the smartest person. I just didn't get into it as soon as I should have, but that's okay. You can always learn things later on. So yeah, cryo-electron microscopy started off as, like I said, this sort of blob-like structures, which you could still learn information from. It was still very useful. But then when these detectors came out, it was just this huge boom all of a sudden, they were like, hey, we can do what you do too. But what's really great about electron microscopy is that crystallography, it can be challenging to crystallize larger protein complexes, like over 100 kilodaltons. Not impossible, people have done it, but a lot more challenging. Whereas cryo-electron microscopy, you can do these large protein complexes, right, and much more rapidly. It still has its own sample preparation challenges like crystallography. But, you just get it and you stick it in the electron beam, for lack of it's not very technical. The robots do it for you depending on the microscope that you have.
Yeah.
And then there's different energy level microscopes you can get access to, I should say. And so some are good for screening to see, will this work? Like, can you see, basically you see a projection. It's like a shadow of your protein. And you need to get different orientations of that shadow to make your reconstruct a 3D structure. Right. So one of the challenges you might see is you get just like all your proteins are in one, like they're all laying flat in one orientation. So then you can say from the screening, okay, I've got to go back and figure out how to put them on this grid so that they show up in different orientations. But then when you do see that, it's really cool that you see these, like these shadow projections, and you can see different features. Like, if you've got something that forms like a hexamer, you can often see that hexameric ring if it does that. And so, yeah, it's just amazing what the technology does now. So now you have all these options available to you as a structural biologist, whether it's crystallography, cryo-EM, or even like AlphaFold, right, to predict your structure.
And your lab is sort of leveraging these technologies to study things like you said, DNA repair mechanisms and different things with bacterial genetics and their differences. And can you just explain what the implications are of the research that you do in general?
Yeah, that's a really good question. So at heart, I love the fundamental science, right? Understanding how things work. But, you can always look forward and see what might this give us. And so looking at DNA repair pathways and DNA damage tolerance pathways, right? Basically, how does a bacteria keep its DNA safe so it can keep replicating and living? The hope is that when we study these pathways, we'll eventually find some proteins that will be good targets that if you inhibit them, or maybe if you make them keep them turned on all the time, they will be ways to kill the bacteria. So it could be potential antimicrobial therapeutics, right? Or potential targets for antimicrobial therapeutic development. So that's one angle. And then a lot of the proteins we study have homologs in humans, and DNA repair pathways are often good targets to kill cancer cells as well. So there's potential as well for cancer therapeutics down the road, which that's another project going on in the lab that I never thought we'd get to, right? I wasn't sure if we get to, but looking at potential inhibitors of our proteins that might have therapeutic potential one day.
And we talked about this a little bit before recording, but I just kind of wanted to ask your thoughts on like AI in research.
Yes.
Because, it's a very prevalent tool, of course, very controversial, but as we talked about before, it has its uses and it has its benefits. So you don't really have to talk about if you use it in your lab, but like just generally, do you think it's a useful tool in research?
I think it is. I think it's fantastic, like what it's made us capable of. So in our, I don't mind talking about what we use it for our research. Like as a structural biologist, we always want the experimental structure, but if we can't get that fast enough, now I can just predict the structure. And that helps us generate so many hypotheses and research questions. And, if we're looking at a protein, we tested it in the lab, we see that it has polymerase activity. Okay, where is that polymerase activity coming from? What's important for that? We can get this model now, make mutations, right, to the protein itself based on that structure and test it to see if that is true or not, right? So normally you'd sort of maybe wait for your structure, or you just take this mutate everything approach, like alanine scanning mutagenesis, to try and find what's going on. So it can help you be more targeted. I think that's amazing. And also now, if you want to find inhibitors to your proteins of interest, so there are a number of different algorithms out there and tools, and one that's quite new is called BoltzGen. And you can give it your protein and say, I want a peptide inhibitor, design an inhibitor. And so it will help. BoltzGen is one. There's also RF diffusion and MPNN. These are all just names of different tools you can use, right? That let you give a protein structure, whether it's experimental or predicted by AI, right? And then it'll give you back a peptide. It says, or give you a bunch of different peptides, and it shows you where it might theoretically bind to your protein. And so then you can purchase that peptide or synthesize it yourself and test it in the lab, right?
That's amazing.
It's just speeding everything up, right? So I think we've got to be careful how we use it, but if you're careful with it and you're very clear in what you're using it for, it can have so many benefits, right? It's going to help us get therapeutics faster. That's not a bad thing, right?
Oh, yeah.
Especially because, it already takes so long to get new drugs out. So if we can kind of speed up the research process, at least we can kind of turn 15 years to at least maybe 10 years or 8 years,
Exactly. I think that's the biggest benefit we're going to see with this.
That's amazing.
Yeah. And you also, what would you say so far amongst your time here at McMaster has been sort of your proudest accomplishment with your research?
That's a good question. So as much as I love the science and the research and I love what we do, the thing I really love and most proud of is the team I've built and the team that we've built together. So the people in my lab, I love when we have we have one-on-one meetings weekly or bi-weekly to discuss research. We have our regular lab meetings weekly, and when there's good discussion going on and new ideas are being generated, I just think that's the most amazing thing. Like, I come out so energized and excited for the research, and then, I have to say, I'm super proud. I graduated my first 2 PhD students this past convocation.
Oh my God.
And so, they both started together in their masters, and seeing them when they first started to walking them across the stage, I had that proud parent moment, and I'm just so excited for them. And so the first 2 is a really big deal, and I'm excited for all the students down my lab. But I think that's what I'm most proud of, is the people and the community we've got for science.
I completely second that. Honestly, I so far during my own time engaged in research and stuff, my main thing that I've been sort of drawn to was just that, the community,
right?
And that feeling when you're bouncing back ideas,
yes,
and you're basically just just throwing darts, and one of them lands, and you're like, oh my God, wait, my idea has merit.
Yeah,
and,
exactly.
Yeah,
or you build off of one another.
Yeah.
The science is wonderful, but it can be really hard. So you need that community around you to support you.
Yeah.
And those people, if you can get through a PhD, graduate school with people, you are bonded for life.
Yeah.
I'm still in contact. I have a WhatsApp chat with, we call ourselves the docs, right? It's the friends that we went through our PhD with. We chat all the time.
Yeah. And you think the collaborative aspect of research is sort of the thing that potential prospective PIs should try to highlight the most whenever they're trying to start their own groups or such?
Definitely. I think that's how you grow and learn. I had the naive idea that science was very siloed when I first started, and maybe once upon a time it was, but now that's not the case anymore, right? If I have an idea, as a scientist, you can read about all these ideas, but maybe you only have certain expertise in your lab for an experimental methodology. And I might have a question saying I really want to test this out in a worm or something. So I'm going to go, if I want to work with C. elegans, I'm going to go to the Health Science Center and find Dr. McNeil and talk to her, right, about what to do with this worm. Or if I have an idea to develop some sort of biosensor with some DNA repair, I'm going to go to Dr. Lee and talk to him about, how can I design this, right? I think it's really cool in our department how we have people with such different expertise that you have, all these, there's also much potential there, is what I would say, right? It's very exciting. I just wish I had more time in the day to sit and think about what I could do. Yeah.
I know. I know we both talked about, collaborations and connections, and I just want to add on to that because especially, we're both in the same lab, we're both in the Lee lab.
Nice.
And, just seeing, we talked a little about this before, me and Dr. Andres, so I don't want to leave you out, but just seeing the connections and seeing the journeys of the master students and the PhD students in the lab is just really inspiring. And just seeing their challenges and trying to learn from their own experiences, I think that's really valuable for us as undergrads who are, trying to find your own paths in life. And, it's really, I'm really grateful that even an amazing prof like you kind of agrees with those ideas and those sentiments. So, thank you so much.
Yeah, you're welcome. Everyone's journey is different, right? So the more you can learn from everybody, it can only help you see what's possible, right?
Exactly.
Yeah. Well, honestly, I think your students, which is us now, who are taking your class, and all of your lab is very lucky to have you. This has been a truly enriching conversation. I honestly learned a lot today, and I'm sure all the listeners out there have as well. Thank you so much, Dr. Andres, for taking the time to share your story with us. And thank you to everyone listening. We'll see you next time on Office Hours Unplugged and maybe have you again in the future.
Sounds great. Thank you much.
Thank you.
Thank you.